Today, we're putting on our detective hats and diving into a fascinating molecular mystery. Our case is all about how one single, tiny flaw in our genetic code can unleash a truly devastating brain disease. We're going to follow the clues right down to the cellular crime scene.
But first, I want you to imagine something. What if you started to lose yourself? Not just your memories, but your personality, the way you talk, the way you connect with people. What if the very essence of who you are just started to fade away? For people with a certain kind of dementia, this isn't a hypothetical, it's their reality.
So what we're talking about here is called frontotemporal dementia, or FTD for short. It's this umbrella term for a group of brain disorders that specifically attack the frontal and temporal lobes. You know, the parts of your brain that are basically the headquarters for your personality, your behavior, and your language. When those areas start to break down, the person you are starts to change.
Now here's where it gets really specific. For a whole bunch of FTD cases, scientists have traced this entire heartbreaking disease back to a single culprit, a faulty gene called GRN. The GRN gene is supposed to make a protein called progranulin, or PGRN, but when the gene is defective, the brain just doesn't get enough of this crucial protein.
And that, right there, is our central mystery. It's the big question that researchers have been chasing for years. How can a shortage of just one protein cause such widespread, catastrophic failure in the brain? To get to the bottom of this, we have to zoom way, way in. We're going to the cellular level. All right, our investigation starts inside a tiny little compartment in our brain cells called the lysosome. The best way to think of the lysosome is as the cell's ultimate recycling and waste disposal center. It's constantly breaking down old, worn-out parts and cellular garbage. And guess what? This is exactly where our protein, progranulin, does its most important job.
So you can probably see where this is going. When that GRN gene is faulty and you don't have enough progranulin, things inside that recycling center go haywire. The whole system gets clogged up, creating this massive cellular traffic jam. Waste starts piling up, and one type of waste in particular, fatty molecules called lipids, really starts to accumulate.
But, you know, lipids is a pretty broad term. To really crack this case, scientists had to get more specific. They needed to pinpoint the exact kind of cellular trash that was causing this massive clog. They needed to find the main culprit.
And they found it. The junk that was piling up was a specific class of complex lipids called gangliosides. Now, this was a huge clue, because gangliosides are all over the place in our nervous system. And we already know from other diseases, like the devastating Tay-Sachs disease, that when gangliosides build up, it's incredibly toxic for the brain.
And the proof? Well, it was undeniable. When researchers looked at brain tissue from FTD patients who had that GRN mutation, they found that the levels of these gangliosides were through the roof. We're talking 2 to 4 times higher than in healthy brains. The traffic jam was real, and it was bad.
But just when they thought they had it all figured out, boom, a major plot twist. I mean, common sense would tell you that if this trash is piling up, it must be because the enzymes that are supposed to clean it up are broken or missing, right? Well, wrong. When they measured those cleanup enzymes, their levels were totally normal. And this just created a much deeper puzzle. Think about it. You've got a huge pile of garbage, and the entire cleanup crew is standing right there, ready to work. So why in the world isn't the trash getting cleared? It meant something else. Some hidden accomplice was missing from the scene.
So the hunt was on for this missing piece. Scientists realized they needed to look beyond the cleanup enzymes themselves. They started wondering, what if the enzymes need some kind of helper to do their job? And that's what led them to a little-known but absolutely critical molecule inside that lysosome.
That molecule is called BMP. Now, BMP is another type of lipid, but it has a very special role. It works as a helper molecule. It's like the special tool that the cleanup crew needs to grab onto the garbage. Without BMP, the enzymes just can't get a good grip on the gangliosides to break them down. It was the missing link.
And this is where the whole case just cracked wide open. The data showed that in cells, in mice, and in human brains, all of which were missing that progranulin protein, the
The levels of this essential helper molecule, BMP, were slashed by about half. The cleanup crew was there, but their tools were gone.
So let's just make this crystal clear. The cleanup enzymes were there the whole time, ready to go. But without enough BMP, they were completely powerless. They couldn't do their job. And so that ganglioside trash just kept piling up and up and up.
So here it is, the whole chain of events, the domino effect. It all starts with the faulty GRN gene, which means not enough progranulin protein. That shortage of progranulin causes the levels of the helper molecule BMP to plummet. And without enough BMP, the cleanup enzymes can't work, which leads directly to that toxic buildup of gangliosides. Case closed.
And this wasn't just a theory, they actually proved it. In the lab, they took some of these progranulin-deficient cells and just added BMP back in. And what happened? The ganglioside levels went right back to normal. As the scientists themselves put it, this proved there was a direct causal link between not having enough BMP and that toxic garbage pileup.
Okay, so we've solved the mystery at the molecular level. That's awesome. But how does a little traffic jam inside a cell lead to the kind of massive, widespread brain damage we see in FTD patients? Let's zoom back out now and connect the dots. See, this problem doesn't just stay inside the lysosome. That buildup of gangliosides acts like a blaring alarm bell for the brain's immune system. It triggers a huge inflammatory response, something we call neuroinflammation. And this kicks off a toxic chain reaction, a cascade of damage that hurts not just the neurons, but all the other important cells that are supposed to be supporting them.
And the damage is just everywhere. The brain's own immune cells, the microglia, go into this hyperactivated state. The astrocytes, which are the support cells, become dysfunctional and can't do their own cleanup jobs properly. Even the oligodendrocytes, the cells that make the insulation for our brain's wiring, start to fail. It's a total system-wide breakdown.
Now, understanding this whole molecular pathway, from the gene to the garbage pileup, isn't just a cool science story. It completely changes the game. It gives us a brand new roadmap for how we can fight this horrible disease. The practical implications here are huge. For one, we might be able to develop new biomarkers. We could track the disease just by measuring levels of BMP or gangliosides. We could also design new drugs that aim to lower the production of those gangliosides. Or, and this is really exciting, we could develop treatments that go right to the heart of the problem and try to restore the brain's supply of that crucial helper molecule, BMP.
And all of this leaves us with a really fascinating final question. We started this journey with a single faulty gene, and we ended up with a problem of lipid imbalance. Could it be that for FTD, and maybe even for other neurodegenerative diseases, the key to a future treatment isn't about fixing the gene itself, but about rebalancing this delicate ecosystem of fats and lipids inside our brain cells? Thanks to this incredible research, that's a question we are finally in a position to start answering.